Okay, so here we are back, second part of the digestive system. We've worked our way through the mouth, through the pharynx, the process of deglutition, and now we are beginning the rest of our little journey. Right, so again, just as a reminder, we started off here passing into the oropharynx, then to the laryngopharynx. There, we're going to have the epiglottis kind of move us back away from the trachea into the esophagus behind it. And the esophagus is unlike the trachea, which is held open by those cartilage rings, the esophagus is flat and compressed at rest. And it's really only kind of pushed open by movement of a bolus into it. And it's going to run from the bottom of the pharynx all the way up, or all the way down, to the stomach, which means it needs to pass through the diaphragm. And so back, if you look at the diagram, if you saw in the models that we had in lab prior to this, there's a little sort of U-shaped cutout on the dorsal side of the esophagus by the spine. That's the esophageal hiatus. That's where the esophagus passes through it. It's going to stop, it's going to terminate at the first of our sphincter muscles. And all a sphincter muscle is, it's a circular muscle that's going to close off passageway from one part of the alimentary canal to another. So here, as we transition from esophagus to stomach, we have the gastroesophageal, gastro here meaning stomach, esophageal meaning esophagus, or the cardiac sphincter, separating those two things out. Now, transport here in the esophagus is via a process called peristalsis. And peristalsis is a very simple process, and it involves kind of the alternate contraction of the circular and longitudinal layers of muscle to squeeze food, in this case our little bolus here, from the top to the bottom. It works very much like squeezing toothpaste out of a toothpaste tube. Okay, and that's really all that we do here in the esophagus. We don't add a lot of extra stuff to it. So it's not a very interesting organ, but it's a very important organ, but it's not a very interesting organ. What is a little more interesting is the stomach. The stomach you'll find in the upper left quadrant, for those of you who remember your quadrants. And its job is, well, it's got a lot of different jobs, put it that way. Primarily here, it's a storage device. It sits in, food sits in here for quite some time, actually. But primarily we think of it as being a site of chemical digestion. Again, we have a typo that wasn't a typo, but I believe my autocorrect fixed that. Instead of chyme with an I, that's chyme with a Y. But what we're doing here is we're taking that solid bolus, right, and we're breaking up molecular bonds, okay, and making it into kind of a slurry or a soup. And that slurry or soup is called chyme. And the food, the rest of its journey until it becomes feces, is going to be referred to as chyme. Again, that's chyme with a Y and not with an I. Stomach is divided into 4 different regions. The cardial part, which is where we're going to begin. Remember the cardiac sphincter, the cardial part of the stomach. The fundus, okay, which means globe or world in Latin. The body, and then the pyloric part is the sort of the tail end of the stomach. One other thing that's nice or interesting anyway about the stomach is that muscularis externa, which normally has 2 layers to it, has a 3rd layer of muscle with that. So here, looking at the stomach right off the bat, we see muscularis externa, our normal layers in here, right? We have the longitudinal layer on the outside, the circular layer that would be the inside layer. We have this 3rd additional oblique layer just inside of that as well. And the reason we have all these different layers of muscle here is that the stomach really grinds and churns and has a lot of very specific movements as it kind of sloshes the chyme around inside the stomach to mix it and to do a little bit of mechanical digestion as well. And so we add this 3rd layer of muscle here to kind of help accomplish that. Esophagus coming in here, the gastroesophageal or cardiac sphincter right there. So we enter the stomach here at the cardia. The part that kind of sticks up like a ball or a dome here is the fundus, okay? And then we have the body kind of in the middle. There's not really a nice hard and fast rule about where the cardiac or the cardia region and the fundus region, the body, all kind of divide out. They're just kind of general descriptions here. And then eventually down to the pyloric part, to the tail end of it, where we have our second sphincter, in this case the pyloric sphincter, separating the stomach from the beginning of the small intestine. The inside of the inner layer here, the mucosa of the stomach, you can see has a lot of texture to it. Okay, these little textured little ridges here are called rugae. And the job of the rugae here is to increase surface area. And the reason that's so important, right, is that the lining of the stomach here is there to secrete gastric juice. And so the more, and we'll talk about what that is in a second, the more surface area we've got, the more area we've got to secrete it, the faster we can secrete it, and the faster we can get about doing the business of digestion. The other bits of important anatomy here for us, we have this smaller, sharper inside curve we call the lesser curvature of the stomach, and the sort of broader outer curvature here we call the greater curvature. These will come up a little bit later on. If we put the stomach kind of in body orientation here, we see that there are two mesenteries holding the stomach in place. We have an anterior mesentery from the lesser curvature to the liver. This is the lesser omentum, okay, and you can see it in all of its glory right here. And then the greater omentum, which is what this little bit here is, unfortunately, in this diagram has been cut. It is this big floppy bit that comes down, covers the entire front of the abdominal cavity. So if the greater omentum was here, attached to the greater curvature of the stomach, if the greater omentum was here, you wouldn't be able to see the intestines. They would be covered by this mesentery. And this mesentery comes down to about here, then folds and comes back up and actually attaches to the large intestine here at the transverse colon. So it's kind of just a big flap that sticks down. Our big sort of adaptation here in terms of functional adaptations has primarily to do with modifications to the mucosa, right? And it's there to make basically two things. Mucus, of course, because we're making mucus the whole way through, but also here in the stomach, gastric juice. Now again, gastric here refers to the stomach, so this is fluid produced by the stomach to be released within the stomach. And I'll just show you the picture for a second. Basically what happens is we have in the mucosa all of these little holes, that we call gastric pits. And these gastric pits dead end in little glands that we call gastric glands. And these gastric glands secrete a couple of different things. Hydrochloric acid is kind of one of the big ones. Enzymes that break down proteins, proteases, okay? In particular, something called pepsinogen, which becomes pepsin, kind of a fairly common, fairly powerful protein digesting enzyme. And also some, not a lot, but some lipases here as well. So it's pretty nasty stuff, right? It's there to break down basically all of the stuff that we're made out of. So in order to keep it from digesting the stomach itself, that's where the mucus comes in for us. And the mucus made by the mucosa in the stomach is a little bit special in that it is very, very rich in bicarbonate. It's very, very alkaline. It is very, very basic. Okay, and because of that, it neutralizes this, a lot of anyway, this hydrochloric acid. Now, it's not perfect, right? It actually doesn't do a perfect job. We still have to replace that mucosa every couple of days, 3 to 6 days, depending on how much you're eating, what you're eating. So we still have to kind of have some turnover there, but it makes those cells last a little bit longer. We look a little bit closer here at the gastric gland itself, and we see that there are 3 different kinds of cells within it. mucus cells and mucus neck cells. You can probably guess what a mucus neck cell makes. And then also we have parietal cells here in blue and chief cells here in the sort of purple color. The parietal cells here are there to produce hydrochloric acid. Okay. The chief cells produce pepsinogen, which in the presence of hydrochloric acid becomes pepsin. And these two things here do a very, very good job at breaking down bonds in both animal and plant proteins. Now, because this stuff is so nasty, we don't want to make it all the time. Right? That would be really bad, honestly. And so we're going to go through 3 phases of kind of control where food is moving, potentially is moving through our digestive system. So we turn things on and turn things off when we need them on, and turn them off when we don't need them. So we're going to begin with the cephalic phase. Cephalic means head, okay? And so this is going to be processes related to the sense organs of the head, okay? So seeing food, thinking about food, smelling food, tasting food, all of these things are going to stimulate the production and the release of gastric juice, okay? Depression here, in the clinical sense, being like clinically depressed, actually inhibits this cephalic phase quite a bit. So even if you see or smell food, you won't produce as much gastric juice as you would normally. The gastric phase here has to do with the stomach, as again, you probably have guessed, right? The food filling the stomach activates stretch receptors in there, chemoreceptors, primarily ones that are sensitive to proteins, but also caffeine will also stimulate production of gastric juice. Being under stress, okay, or if the acidity of your stomach contents gets too high, both of those things will inhibit the production of gastric secretion. And then in the intestinal phase, right, so we've begun by, okay, we want to turn it on and get ready for food. Food is here, we need to make it so we can digest it. Now we're moving that food, that chyme here, into the intestines. So we're starting the intestinal phase. As we fill the first part of the small intestine, the duodenum, this is a trigger that says, okay, we're done digesting, food is moving on, let's turn off the tap. However, depending on how completely digested that food is or is not, it might be a cue to say, hey, we're not quite ready for this yet. Turn the gastric secretions back up, stop pumping it into the small intestine, break it down a little bit more. One thing we've talked about before with smooth muscle here is this notion of sort of a stretch relaxation response. Okay, here in the stomach, we refer to this as gastric accommodation. Basically, the idea here is that your stomach is going to stretch, right? It's empty, right? Start to fill it up to about 1.5 liters of food. Okay, you're going to that should be an L in there. I don't know what happened to it, but 1.5 liters of food. We don't really do anything. But as we go beyond that, now we're going to get what's called this receptive relaxation response. Basically, the idea is that we're pushing the stomach beyond its normal limit, and we're going to reset that relaxation point, right? So we can have full strength contractions at that stretched position. But of course, once all the food is moved on, we go back to normal. You don't permanently stretch your stomach out. Nothing like that. So the process in here and why we need those 3 layers of muscle is kind of a 3-step thing. It's going to involve peristalsis again, okay? But peristalsis here is going to move our chyme not down and through an open sphincter, but actually against a closed sphincter. So as we have a wave, right, through the body, through the pyloric part, we're going to kind of splash this food against this pyloric valve. And as we do that, it's going to rebound, right? It's going to rebound, it's going to splash up here against and up into the fundus and come crashing back down. Right? And we're going to do this and do this and do this and do this and do this again and again and again and again. This is called propulsion. And as we get closer and closer to the pyloric part, we get more and more kind of vigorous peristalsis, we call that grinding. So propulsion and grinding are kind of activities occurring at the same time. It's the same activity, it's just a different level of intensity. The splashing here, right, this return from this pyloric valve is called retropulsion, right? And again, this is just about trying to mix all of the food, all of the chyme in here to try to get it into contact with all of those digestive enzymes, all of those things we're trying to break down so that we get a nicely digested meal before we emit a little bit, a couple of mils at a time, into this first part of the small intestine. Which brings us to the small intestine. There's three chunks of this in order. They are the duodenum, the jejunum, and the ileum. Of these, the duodenum is our first retroperitoneal alimentary canal organ. Okay, now there's only been two that could qualify, the stomach and the duodenum, but it's still the first. Small intestine is primarily here for absorption, but it actually does a fair amount, particularly in the duodenum, of digestion as well. It's anywhere from about 5.5 to 6 meters long in the average adult. And as we get farther and farther along, each of these parts gets a little bit longer. So the duodenum is somewhere around 25 centimeters long. The jejunum is somewhere around 2.5 meters long. And the ileum, the last part, is somewhere around 3.5 meters long. And these are these parts here. Now, the duodenum is easy because the duodenum is this little C-shaped part right here. And usually, and I wish I could show you this in person, it's stiff. It's got a lot of cartilage in it. If you squeeze it, it actually crackles. It makes a little crackling noise. And it's the reason it's stiff and it has this very specific shape is because it's going to receive a lot of chemicals from outside the alimentary canal. And so we want it to be rigid so the connection point doesn't get pinched off or otherwise improperly dealt with. The jejunum and the ileum, on the other hand, if you just see them, if you just see them kind of from this view, you'd be really hard pressed to tell them apart. Really, the big difference is going to be histological, where we see the ileum here will have those Peyer's patches in it. You can feel them. If you have the intestine in your hand, you can feel them. If you have a histological section, you can obviously see them, but otherwise they're really hard to tell apart. Okay, so the duodenum, which is going to be this guy right here, we've got a lot of stuff coming in, additives to the chyme at this point. So within the duodenum itself, we have Brunner's glands, and Brunner's glands or Brunner's glands secrete mucus, as we've seen before, but it's very alkaline. Right? Again, when we think about what we're dumping in here, it's chyme, right? But it's also a lot of hydrochloric acid. The stomach has its own mucus neck cells that produce alkaline mucus to protect it. The duodenum doesn't have any of that. So the duodenum has to have these Brunner's glands here to make this alkaline mucus so that it doesn't burn a hole, literally burn a hole through the duodenum as it passes its way through. Because it can sit in here for actually quite a long time. You wouldn't imagine it to be so, but it really can sit there for a long time. The other, again, I don't know why this autocorrected to chyme every time. That there is a Y there. I don't know what's going on with that. It's going to receive chyme across that pyloric valve. It also receives bile through the bile duct from the liver. And the role of bile here is to emulsify fats, and we'll talk about what that is really more in the next chapter. We're also going to receive pancreatic juices, of course, from the pancreas through the pancreatic duct. So this would be, again, stuff that's going to be alkaline, so bicarbonate, some amylases here to break down carbohydrates, some lipases to break down fats. The bile duct and the pancreatic duct merge, and they actually enter the duodenum together, something we call the major duodenal papilla. So again, we go back to this little cartoon here. Here's our pancreas nestled in this little crook here of the duodenum. Okay, the pancreas is kind of an interesting organ. You can see here this cartoon doesn't really do it justice. it's very much got the texture and consistency of chewed bubble gum. It's one of the few organs in your body that doesn't have an outer membrane or an outer lining or an outer covering. And so we don't have a nice smooth surface to it. We have just sort of the inner parts to it. And it doesn't have one because it actually sits inside the mesentery in this part of the body. So it doesn't need an outer membrane. But it's going to produce various things here. We'll talk about what those things are in a little bit. And dump here into this little conjoined passageway with the bile duct coming down from the liver here at this major duodenal papilla, which is this guy right there. The jejunum and the ileum are really going to be highly adapted for absorption. This is really mostly what they're doing here, okay? And so when we look at adaptations here, again, like the stomach, primarily to the mucosa, only now, right, we're going to have this not for secretion, but for absorption. And so we're going to go kind of in order here, and we'll see a cartoon on the next slide of what this looks like. These circular folds are plicae, and then villi on each of those plicae, and then microvilli on each of those villi. So here's a chunk of, say, the ileum or the jejunum. I suppose it doesn't really matter. I don't see any Heyer's patches, so it's probably jejunum. For these big structures we're seeing here, right, these are those circular folds, or the plicae. And then if we take a little cross section here through one of those folds, you can see that there's also all these little fingers sticking up out of it. Those are the villi. And if we zoom in on one of those villi, okay, so here's our villus here. We zoom in on the outside of it. Here we've got all these tiny little fingers on each of the little cells of the villi. These are the microvilli. And they form what we call the brush border. Okay, and that brush border has massive surface area. So as food passes through here, we've got a lot of opportunities. As chyme passes through here, a lot of opportunities to encounter a nutrient and to absorb it, to bring it into the body. At this point, the chyme, finally, mercifully spelled correctly, contains a lot of partially digested stuff. Okay, carbs are mostly broken down, proteins are mostly broken down, fats not really broken down really at all. And so food is going to spend somewhere around 3 to 6 hours. The more fat that's in the chyme, the longer it's going to sit. Being sort of finally digested all the way out, right? So things like nucleic acids, these partially digested proteins, fats, finally get broken down into smaller chunks and absorbed. And really, again, this is the primary job. So fats, amino acids, carbohydrates, ions, electrolytes, right? Water, most vitamins, A through E, vitamin K, also get absorbed here in the small intestine. Transport, our old friend peristalsis returns, right? This is how we move food from one end of the small intestine to the other. But we also do a lot of mixing here. And the movement that causes mixing is called segmentation. So rather than this rhythmic squeezing that starts here and pushes food back this way, what we get is rhythmic squeezing where we kind of slosh food back and forth, right? Kind of mix these two chunks again and again and again and again and again. And the reason for all that mixing is that mixing has the effect of bringing it, more of it into contact with that brush border. Remember, the process by which we're getting food into the body from the chyme is largely diffusion. And if it's not diffusion, it's active transport. And both of those things take they require very, very short distances. So if the nutrient isn't within basically contact distance of that brush border, it's not going to be absorbed. So by churning and mixing and segmenting, we give ourselves a better opportunity to pull those nutrients back out of it. Eventually, we're going to meet the large intestine at yet another sphincter. Here, the ileocecal sphincter, because we are in the ileum, and the next part of the other side is called the cecum. The ileocecal sphincter is the last part of our small intestine. It is going to open, not like the pyloric valve in response to kind of being continually splashed against and allowing little spurts through. It's going to open in response to what we call the gastroileal reflex. Gastroileal here, again, we get some signal from the stomach as we eat food. If there's still food in the small intestine, move it along, okay? But also if food kind of chyme at this point bumps up against our ileocecal sphincter, it'll pass it along through there. It's not going to sit and wait at that gate. Okay, so we'll come back. We'll do the final part of this with the large intestine and the rest of kind of the accessory organs, and then a little bit about various things that can go wrong.